Okay, welcome to podcast 37. We are looking at either classical or hereditary or Mendelian genetics, whatever you want to call it, but the traditional form of genetics study, and where we look at how traits get passed from one generation to the next and what these things we call genes actually look like when they're being expressed in a living thing. And everything that we're studying in this unit got its start with this man that you're seeing on the screen. Of course, he's Gregor Mendel, and I'm sure you studied him in your first year of biology and maybe even back in middle school. He's the one that gave us the laws and concepts about genetics that we're still using today and that we're going to be learning about in detail now. Hopefully this will be a refresher. What I want you to take away from this podcast are these main topics. Okay? First, we want to know what this man did, and we particularly want to know how it was different from what other people were doing at the time when he was doing his work. We want to understand how to do a monohybrid cross, which I hope you already do, a dihybrid cross, which I'll also bravely hope you already know about that, or will take only a little bit of refreshing, and how it is that the chromosomes, what they are doing, actually relate to what we see in these crosses of plants or people or other organisms. Okay? This is where Gregor Mendel worked. He was a monk in an abbey in Austria, and this abbey, many of the monks in this particular abbey had an interest in science, and many of them pursued scientific endeavors. He was not the only one. He was not an oddball while everybody else was out doing other things. There were several monks there that did plant breeding experiments like Mendel did, but obviously he took his quite to the extreme in one direction into this area of genetics. Today his garden looks like this. They've preserved it where he did his work, and you can see a little historical marker over here. There's a statue of him on the grounds as well, so the abbey still exists, and his garden place still exists as well. Now, what he did that was really so different was his careful use of what we would recognize today as good experimental method. He controlled his experiment. He didn't just... The typical genetics experiments being done at the time, first of all, we're looking at humans. Now, that just does not work. Humans don't cross with whoever you tell them to cross with. They don't breed with who you wish for them to. It takes for 9 months just to get a baby, let alone to necessarily see how a trait is going to be expressed in that child by the time it gets to be an adult. Humans are horrible subjects for genetic experiments, yet that's what most of the genetic experiments were using at the time. They would look at a trait in a couple of family groups and how is beauty being passed from one generation to the next. Horrible, subjective, poor experimental design. He had excellent experimental design. So he was used excellent experimental method, controlling his experiment in terms of what variables were going to be allowed and what was not, and so on. Additionally, he used quantitative analysis. Most experiments that were being done at that time used qualitative, subjective, this person's pretty, this person's not, whatever, and only had a few subjects to analyze. He used quantitative analysis where he counted his pea plants. How many showed purple? How many showed white? How many were tall? How many were short? Thousands of them. You can see some of his work in statistical analysis here. He would count them and then analyze them statistically. He did really careful work that allowed him to perceive trends that he might not have perceived without this mathematical basis. So He just did his work so much better and more carefully than anyone else in his time. Additionally, he worked with pea plants, and for pea plants, there are many traits that are what we would call discrete, meaning they are either on or off. The plant either shows the trait or it shows another trait. There is no in between. The common idea for traits during Mendel's time was an idea called blending, that if you, for example, in paint, if you have red and you have white, you put them together, you get pink. It's a blend. Well, this was what was thought to happen with regard to traits. You have tall and you have a short, you get a medium. That's two parents. Their child is a blend of their traits. Well, obviously there are traits that just don't work that way. We'll hope that you're not a blend of your parents' genders. You are either male or you are female. There is not blending in a normal situation. So blending left many questions completely unanswered. And so it should have been a pretty big tip-off that that wasn't it, because pea plants so clearly show non-blended, discrete traits. This was an excellent choice that Mendel made. He followed about 7 traits at one time or another. Purple versus white flowers, yellow seeds versus green seeds, round versus wrinkled, and so on, tall versus short or dwarf plants. And these traits were either one way or the other. They were never a blended form. A very lucky choice that Mendel made. So, how did Mendel do a cross? This is what he did. He would start any cross with 2 parents, because this is sexual reproduction we're talking about, and they would be true breeding, meaning that he would, well, let me first, before I talk about what true breeding means, let's talk about how pea plants normally breed. Let me go to my next slide. This is what a pea plant flower looks like. The flowers are the home of the reproductive organs, the sexual organs. And in pea plants, plants have both male and female reproductive parts in the same flower. Now, we can't see them in this flower, and that's important. They are hidden inside these flower petals. You have to pry the flower petals open to see the male and female reproductive parts. If you pry open those petals, right here I'm seeing, looks like 1, 2, 3 male reproductive parts. I really can't see the female part in here, but they're all there. What this means is that this plant normally self-fertilizes. Normally, it is an asexually reproducing plant. It self-fertilizes. It clones itself over and over again. That meant that Mendel had excellent control over what happened with his plants. He would start with true breeding parents, meaning he would if he had a white flowered plant, he would watch that plant and observe over generations, letting itself fertilize. Did it always produce white flowered offspring? If it did, he knew it was true breeding. He would let it produce generation, and if he never got any other flower colors, he knew it was pure for that trait. Likewise with the purple flowers, he would watch it generation after generation of self-fertilization. Did it always produce purple flowers? If yes, he knew it was true breeding for that trait. Why was this so important? Because it gave him control. It controlled his experiment. He wasn't going to have weird other color variations popping up in offspring that weren't a direct result of a cross that he was going to intentionally make. So these pure, true breeding parents, he would call his P or parental generation. He would cross them. The way he would do that, let's see, the way he would do that would be literally to take the male parts of one plant. Okay, here we go. We've got the male parts right here. This is the female part, the pistil, right there. Okay. Now, he would basically yank the male parts out of a plant. Sorry, fellas, that's what he'd do. He'd just take them out. And he would also, for example, if he's crossing white with purple, perhaps he would yank the white male parts out of this white plant. He'd also get rid of the male parts out of the purple plant. But he'd take the male parts from the white and he would dab the male pollen from this anther right here onto the top of the female reproductive part. So here's this female reproductive part, this vase-like structure, okay, that we call normally a pistil, okay, I can't read, there it goes, a pistil. He would dab the male parts with their pollen grains on top of the pistil, on the stigma, okay, and it's sticky, it's got a little sap-like stuff, and the pollen sticks. Inside each pollen grain, I'm showing you a big old pollen grain, there is a little cell, and that is a sperm. So inside the pollen is sperm. A pollen tube would grow down into the female reproductive organ, and a sperm would move, crawl, kind of like an amoeba, down the pollen tube. In the base of the female part is the ovary. Right here, this is the ovary, you can, whoops, right there. And in the ovary is a structure called an ovule. Actually, there's lots of these ovules down here. And inside each ovule is an egg. So the sperm has to crawl into an ovule where it will fertilize an egg. And once fertilization happens, then this ovule is going to become a baby plant, okay, which we would call a seed. Essentially what happens is that after fertilization, after the sperm from the male part comes crawling down the tube and crawls in there and fertilizes the egg, the flower petals die away, the reproductive parts, the female reproductive part, fundamentally, this part of the flower changes shape and it starts to look like that. And it's the ovary, okay? So if you've ever eaten a pea pod, you are eating an ovary from a pea plant. What's more, you're eating what's inside it, which are the babies. These were the fertilized eggs in those ovules. This is the seeds that were created at the fertilization event. Well, Mendel wouldn't eat them. He would plant them and let them grow. And he would see what kind of plants they produced, what color flowers did they have, and so on. All right? So, what Mendel would do then is he would take the male parts, fertilize the female parts over here, take these male parts, fertilize the female parts over here, so they're crossed. He would cover those petals back up so nothing else could fertilize them, and then he'd wait for peas to be formed. He'd plant the peas to see what kind of offspring he got. And what offspring he got were his children of the parents, what he called his F1 or first filial generation. F1 is the parents of the children. And what he noticed when he did this particular cross was that he would get all purple flowers. The white trait was just gone. It was gone. Where did it go? What he would do next is let the offspring fertilize themselves, basically cross with themselves, fertilize themselves, and then he would get children of the children, the F2, and he'd see what kind of offspring he got in that generation. Okay? So, what he tended to see was the following, and hopefully you'll remember this. He always saw, when he crossed a pure breeding purple with a pure breeding white, he saw all purple flowers all the time, 100%. The white was gone. What did he see when he crossed these with themselves? What he saw when he did that was that the white trait came back, but only in the 25% of the offspring from this cross of this generation. 75% were still purple flowered. And it was always in this ratio, 75 to 25 or 3 to 1. What was going on? First of all, why did the white trait go away here? Why did it come back here? And why was it always in this ratio? Lots and lots of questions to answer. Okay? Now, what's important is that you understand that the reason it turned out like that has everything to do with the chromosomes and what happens during meiosis. Well, first, when you get a set of genes from your mom and a set of genes from your dad, and then what happens during meiosis. So, let's look at pea plant chromosomes. Okay? Here is a chromosome. This is a pea plant set of chromosomes. Remember, you get one set from your mother and a one set from your father. You get these homologous pairs. Okay? Well, on each chromosome there are many genes. Maybe we've got a gene for height that occurs in this position. Maybe we've got a gene for pea pod shape that occurs at this position. Well, up here in this position, flower color is always what occurs. But it doesn't necessarily always have to be the same flower color. In this case, we see that mom sent a gene for purple flowers and dad sent a gene for white flowers. Okay? It's in the same position on the chromosomes. It's in the same what is called locus or position on the chromosomes, but they're different versions of the trait. Okay? We call these different versions of a trait or a gene, we call that alleles. A purple allele and a white allele version of a trait. Okay? And these different alleles for flower color always show up at the place they're supposed to, at the locus they're supposed to, but they may not be identical. Okay? So, for each characteristic, an organism then inherits, for every trait, an organism inherits two alleles, one from mom, one from dad. Okay? Why? Because each organism inherits a set of chromosomes from mom and a set of chromosomes from dad. And they are similar. They code for the same kinds of traits, but not the same exact variety of each trait. Okay? Diploid organisms, which pea plants are, which you are, get a set from mom of chromosomes and a set from dad. So you have a pair of chromosomes, like you have a pair of shoes, one from mom, one from dad. Okay? Two sets of chromosomes, and we call them homologous. This is a great analogy. It's like having two editions of an encyclopedia. Maybe mom sent you the Encyclopedia Britannica, maybe dad sent you the World Book or whatever this is, Encyclopedia Americana. They've got the same basic kinds of information, but not exactly the same. And it's a whole lot of information. Okay? We'll talk about the advantages of being diploid later. So, what Mendel's findings meant. He noticed that some traits seemed to mask others. Purple and white one seemed to be more potent, more strong. Okay? They weren't equal. You didn't get purple and white and get a light purple. You got either purple completely or sometimes completely white, but purple more often seemed to mask the white. He called it the dominant allele. Now we know what that means is that dominant allele often is what is making the functional protein, though this is not always the case. We're going to talk about this later on. The recessive allele very often is making a protein that's not working. For example, in this case, this purple allele is the working protein. This is a broken pigment protein that makes no pigment, hence we see white flowers with that. Okay? So, in this case, we have a functioning allele and a malfunctioning allele. Alright? In this case, this one working allele makes enough of the purple protein to make the plant look purple, even if it's not. So entirely purple. So let's make sure we understand some words. Genotype versus phenotype. Phenotype is how an organism looks. Does it look purple? Does it look white? The physical appearance. Genotype refers to the description of an organism's genes that it has. Its letter combination, if you will. Is it dominant? Is it recessive? And all of that. Okay. We represent alleles as letters. Hopefully you'll remember. Generally speaking, we use the first letter of the dominant trait. So in this case, for flower color, we'll use big P because that is showing us pink is the dominant trait. So we use big P, and little p will represent being white. So a true breeding purple flower is big P, big P. It had only big P's. It was pure for that trait. One big P from its mother, one big P from its father. Okay? True breeding white flower would be little p, little p. And when we cross them, we get offspring that are big P, little p. Why? Because at meiosis, it is determined. Remember, the chromosomes split. We cut the chromosome number in half. So we're going to cut these alleles apart. We're going to cut the homologous pairs apart. And each sperm, if this is dad, he's going to produce sperm. But each sperm can only get one big P. One big P. They cannot carry two to the offspring. Same with mom. Her eggs can only have one little P. One little p. If Mr. Sperm fertilizes an egg, there's only one option. There's only big P's meeting little p's, so the offspring all have to be big P, little p. So what we see here then is that the phenotypes, purple and white, true breeding, are crossed. Their genotypes are big P, little p, and this is the offspring that results. They all have to be big P, little p, because all of them are going to get one big P from the father, one little p from the mother. Okay? Now, what we're going to do here is allow these guys to cross within themselves. So we're going to cross one big P, little p with another big P, little p, and that's going to change things up a little bit. Okay? What we're going to see is that this time, if this is going to represent the male, he can send in his sperm either a big P or a little p. Okay? And the same goes for the female. She can send either a big P or a little b. She can't send both at once, but she can send either or. That means what we're going to see is there is a 50-50 shot that dad will send a big P to the offspring, and so will mom, and that offspring would be big P. But there's also a 50-50 shot that mom will send little p when dad sends big P, so the offspring would look like that. But there's also a chance that dad will send big P and mom will send little p, so we get it that way. There are two ways to get this heterozygous individual, if you will. Or there's a chance, there's a 50-50 shot that dad will send a little p while mom also sends a little p, in which case the offspring would show up like this. So what we end up with then phenotypically is that this one looks purple because that's all there is. This one looks purple because big P dominates the little p. This one looks purple, but this last one looks white. There's no working big P gene. They're both broken, so we see the white flower. Okay? What we have then is 25% that are big P in our F2 generation, 50% are big P, little p, and 25% are white. Okay? But when we see them phenotypically, what we see is these all look the same, even though genetically they're a little different. we see 75% being purple and just 25% being white. So the ratio genotypically, how many big P, big P's, big P littles, little littles do I have? I have 1 to 2, 2 here, 2, 1. Okay? But phenotypically, my ratio is 3 to 1 because these three all look the same, so there's 3 of those for every one of those. Okay? So yes, genotype and phenotype can result in different ratios. Okay? Remember these words. Homozygous means we have the same alleles in the individual, big P, or little p, they're both the same. Heterozygous means we have an individual that has 2 different alleles, big and little. Okay? Homozygous dominant means obviously big. Homozygous recessive means little. Heterozygote is the big little combination. Okay? Now, I think we will stop it there because this has gone on pretty long, and we'll finish this up with another podcast. Thanks for watching. I'll catch you in the next one. Take care.